Are you ready for more, laddie? Hopefully by now you've viewed energy reactions in the cell part 2. Well, this is energy reactions in the cell part 2A, the continuing saga of photosynthesis. We like to call this more fun with photosynthesis. Now, even though this is going to be extra credit for our regular biology classes, if you're an honors level student or an AEP student, you might want to go ahead and view this. It will go ahead and fill in all the blanks and all the things that you really need to know to completely explain photosynthesis. Hey, the learning objectives are pretty much the same as the lesson objectives for the last video. You still have to know where everything is or where everything happens, the important reactants and products, and describe everything. The difference here is that you have to go into a little bit more detail. Actually, for full credit, you have to go into a lot more detail. Now, some things don't change, and there's no real additions or changes. For example, photosynthesis is as it always has been. Process by which light energy is used to convert CO2 and water into useful food molecules. When we start looking at the equation, though, we notice some differences here. The previous equation we gave you was referred to as a summary equation, kind of a shortened version. Here, well, we're still using light energy. We're still looking at 6 CO2s, but note here in the reactant side, instead of 6 waters, we've got 12 waters. And that's a little bit different. Turns out that you actually need 12 waters to make a glucose molecule. You'll see why in a sec. You end up with just a single glucose molecule. You end up with some oxygen as a waste. But note here on the product side, you also have 6 waters. Well, if this were a math equation, we could subtract 6 from this side and 6 from this side, and we'd still have an equivalent equation. But it doesn't exactly show you what happens as far as the chemistry goes. You'll see in just a sec. Anyhow, still looking at light-dependent reactions and light-independent reactions. Those light-dependent reactions do indeed occur in the grana. We didn't lie. You still need light energy to split apart water. You're going to need some NADPs and ADPs. Right? Water is split, and you end up with hydrogen ions, electrons. Oxygen is a waste. The high-energy electrons are actually used to form ATP in a really cool fashion. We'll show you that. And the hydrogen is indeed carried off by NADP to form some NADPH for the rest of the reactions in the light independent side, those Calvin cycle reactions. Okay, this diagram is going to be a lot more detailed than the other one that we had. Imagine this structure as a thylakoid, one of those little discs that are going to be containing chlorophyll and where all the light independent reactions are actually occurring. So if we look at the membrane of a thylakoid, most of it is made of phospholipids, just like a cell membrane. And just like a cell membrane, the membrane is going to be composed, the thylakoid membrane is going to have transmembranal proteins embedded in that phospholipid bilayer. Well, 2 of these are going to be really important because they're going to be containing chlorophyll. They're going to be having the light harvesting complexes within their matrix. And they call these things photosystems. And as it turns out, we're going to identify one as photosystem 2, and the other one over here as photosystem 1. Why did I mention 2 before 1? Well, turns out that when they first discovered these things, they didn't know quite what they did, and then photosystem 2 was discovered after photosystem 1, and we're just kind of sort of stuck with the names. So, deal with it, dude. Other important transmembranal proteins here involve what are called electron carriers. And I'll just label them EC. And they got their own names. This little puppy is called plastiquinone, and this one is ferredoxin, and over here is plasticinin. You don't have to know their names. The function is important. They basically shuttle electrons and carry electrons back and forth. Okay. This little puppy right here, okay, is a very special one called a cytochrome complex, and I'll just label it CC for cytochrome complex. But it actually acts as a proton pump, meaning it's going to actually shuttle protons or hydrogen ions across the membrane. You'll see it in action here in just a sec. In the corner, way over here, we got another important one called NADP reductase. Why reductase? Well, turns out that if you have a reaction where a molecule accepts electrons, it's called a reducing reaction. So a reductase is an enzyme, a protein, which is going to go ahead and cause a molecule, in this case NADP, to accept electrons. It might be a little bit easier to see a little bit later here. One last one, and that's this little puppy down here. This is a special one. It's a special channel protein called ATP synthase, and stuck in the little channel is what is essentially, it acts like a small rotor. And if you can imagine a flow of hydrogen ions going through this channel, spinning the rotor, essentially changing mechanical kinetic energy into electrical energy and then eventually chemical energy, because it's going to cause ADP to accept the phosphate and become ATP. Let me show you this thing cleaned up a little bit. Okay, there's that same thylakoid with the items kind of labeled a little bit more neatly so you can see what's going on. You might want to refer back to this particular slide. It will, give you an idea of where things are, so if you get lost, you can always come back here and figure out where things are and what they're doing. All right, ready for the full story? All right, here we go. Remember, we're going to use light energy to go ahead and break apart water. But here we're going to be worried about exactly how much water. We're going to be playing molecular bookkeeper here. So we said that in our big equation, we're going to put 12 waters in here. Well, light collected by photosystem 2 here is going to break apart that water, and when it does, we're going to end up with 6 O2s, 6 oxygen molecules, which are going to exit as a waste. We know that happens in photosynthesis. But we also know that we're going to end up with hydrogen ions. In this case, how many? Well, 12 H2Os. That's going to give us 24 hydrogen ions. And, as it turns out, 24 electrons. Okay? So I'm just going to put E here, but hey, remember that there's 24 of these little puppies, and I'll put the electrons over here because they're going to eventually get picked up by that electron carrier. Okay, cleaned up the diagram for you and relabeled some stuff so it doesn't get too messy. But pick up where we left off. We've got our oxygen over here as a waste, and we've got these hydrogens. And think about it. Now we've got all these hydrogens on the inside of the membrane here, and that would mean a low concentration outside over here. So naturally the hydrogen is going to move from high concentration to low concentration. Sound familiar? Well, you can't let this happen through simple diffusion because it's a charged particle. Remember, charged particles can't get through that phospholipid layer without the help of, one of these carrier proteins, ATP synthase. So we have a channel which will allow hydrogen to escape from the inside to the outside, going from high concentration to low concentration. And eventually you end up with hydrogen ions on the outside. Well, here's the really cool thing. Here you've got a charged particle moving from point A to point B. It almost sounds like electrical energy, and we can think of that as electrical kinetic energy. But stuck in that little channel protein, that ATP synthase over here, is what is in essence a rotor. It's actually physically moves. As it moves, it creates another form of energy which allows us to convert ADP and its phosphates to ATP. Mega cool. The only problem is this is going to function only as long as this flow continues. But eventually we're going to have an equilibrium develop between the number of hydrogen ions on the inside of the thylakoid and the outside. And if you're not going from high concentration to low concentration anymore, the flow just kind of sort of stops. That's where these proton pumps, the cytochrome complex, comes in up here, because when it is activated by those high energy electrons right here, we can now pump hydrogen from lower concentrations back to higher concentrations. It's got to be active transport, got to have some energy. You always need energy to go from low concentration to high concentration. Where is the energy coming from? Yeah, from those electrons. And as long as that proton pump is operational, we can go ahead and maintain a higher concentration of hydrogen here on the inside, a lower concentration on the outside. This flow continues, and we can go ahead and take up to 18 ADPs to make 18 ATPs. Awesome! Well, we got some ATPs, and we're going to be using those in the light-independent reactions. That's cool. But remember, we have to go ahead and shuffle the hydrogens over to the light-independent reactions, the Calvin cycle reactions as well. So we're going to have to do that, but we're going to need some help. The electrons that we talked about over here, which were using their energy to power this proton pump, the cytochrome complex, now have lost a lot of that energy. So they're picked up by a second electron carrier here and shuffled off in contact with photosystem 1. Photosystem 1, its job is to go ahead and accept light energy, not to break anything apart, but to go ahead and re-energize the electrons, bring them to a higher excited state, and a third electron carrier is going to go ahead and carry it over to NADP reductase here. And NADP reductase, its job is to go ahead and convert NADP into NADPH, but I don't know if you guys remember in that first video, NADP is positive, and hydrogen ions are positive. So how do we get a positive NADP to pick up a positive hydrogen? Well, you got to give it 2 electrons. But this is cool. How many electrons did we have way back here? 24 of them, because there were 24 hydrogens. So what's really cool here is that, hey, if we give NADP 24 electrons, we have enough to convert 12 NADPs into 12 NADPHs. And hey, we're going to need those 12 hydrogens to make glucose, C6H12O6. Pretty cool. Hey, and here's a cleaned up version. We are where we want to be, but not quite, because we've got some mystery atoms missing. Over here, we're shuffling off our 12 hydrogens. But way back here in water, we had 12 waters, which actually had 24 hydrogens. We're missing 12 hydrogens. Now the big question becomes, what do these 12 hydrogens do? Where do they go? Are they important? Are they just eliminated as waste? What the heck's going on with them? Well, for that, we have to go ahead and see what's happening in the light-independent reactions, the Calvin cycle reactions. So, hey, our ride isn't over yet. Hang on. All right, the light-independent reactions, Calvin cycle reactions, same thing. We're going to give you a little bit more detail. Some things don't change. Hey, it still occurs in the stroma. We're still going to need ATP energy to make this happen. We're going to need our hydrogen carrier to build a glucose, so NADPH is going to be involved. And we're going to need a carbon and an oxygen source, because if you recall, remember we got rid of all of our oxygen in the light-dependent reactions. So ATP is still used as an energy source. It's going to break apart CO2. It's going to help build the glucose. We're still going to use hydrogen from NADPH. And we're going to end up with C6H12O6 when we end. But there's a bunch of steps along the way. Okay, I can't show you a pretty diagram of thylakoids and stuff because remember the Calvin cycle reactions are occurring out in the stroma. So there's really no structures to show you. But there are a bunch of new molecules we're going to be talking about. So I'll be doing this as kind of a diagrammatical formula, and hopefully you can follow along as we go. I'll mess things up, but then I'll clean it up as we go step by step. So out there in the stroma, waiting for all of the other ingredients, are a bunch of molecules called ribulose bisphosphate or ribulose biphosphate. I just have them labeled here as RUBP. And a little 5C down here means that it's a 5-carbon sugar compound, biphosphate or bisphosphate, because there are 2 phosphates attached. And I'm not going to show you the chemical formula. That's not super important. But we want to keep track of carbons here and eventually phosphates. So note that we've got kind of sort of this arrangement down here with what is in essence 5-carbon compound with 2 phosphates on the end. Now there's 6 of them, so we end up with a grand total of 30 carbons, right, and 12 phosphates. All right. Well, all this is enzymatically controlled, so there is a really cool enzyme called Rubisco. Sounds like some type of delicious snack cracker or something, but Rubisco kind of sort of controls this first end of the reactions. And what it does is it takes carbon dioxide, and this happens in a couple of steps, but I'm going to combine it into one step so that it's just easier to deal with. And we're going to eventually make a glucose out of these carbon dioxide molecules, so we definitely need 6 of them, right? But what Rubisco does is crack the ribulose biphosphate down and makes it accept the carbon and then converts it into a 3-carbon compound, each with a phosphate on its end, called phosphoglycerate. I'm just using an abbreviation there. Well, we had 30 original carbons. We added 6. Now we got 36 of those carbons. So that gives us enough carbon to make 12 PGAs, 12 phosphoglycerates, right? Or 12 of those 3-carbon compounds. And we're going to, like I say, need those carbon to make a glucose, but remember that the formula for glucose is C6H12O6. And yeah, this will give us our 6 carbons, and the hydrogens are yet to come, but we only need 6 oxygens in a glucose. Over here, got 6 times 2 is 12 oxygens, more than we need. What the heck are we going to do with the extra oxygen? Well, here's the cool thing. Remember those 12 extra hydrogens that we had at the end of the light-dependent reactions? Well, here's their job. You've got 12 hydrogens. You've got 6 extra oxygens. Hey, isn't that enough to make 6 waters? And that's exactly what happens. The 12 hydrogens that were put in from the light-dependent reactions combine with those extra oxygens, and now we get our 6 waters as a waste. If we go back to the expanded version of photosynthesis, we said that we needed 12 waters going in. We got 6 waters going out. That's them right there. Aren't you so glad you asked? All right, as promised, to clean that up a little bit. Hopefully that's a little bit neater. We're down here with our phosphoglycerates, our 12 phosphoglycerates. And each of those is a 3-carbon compound. And you might be thinking, well, hey, 3-carbon compounds, that's like almost like half a glucose. And you're on the right track, but these PGAs, these phosphoglyceraldehydes, although they have enough carbon and they have enough oxygen, they don't have enough hydrogen to make a glucose. But lurking around from products from the light-dependent reactions, remember we have those 12 NADPHs, and they've got our hydrogen. All we have to do is get each of these PGAs to accept the hydrogen. That's going to take some energy. Where does the energy come from? Hey, where does the energy usually come from in cells? ATP. We made ATP in the light-dependent reactions as well. But we don't need all 18 ATPs. We only need 12 of them. Why 12? Well, dude, you got 12 PGAs, you got 12 NADPHs. Why not 12 ATPs? So we're going to spend this energy. Of course, this is going to become ADP and its attendant phosphates, right? To go ahead and strip off the hydrogen, join it to the phosphoglycerates. Of course, you end up with, over here, just NADPs, but that can be also recycled back into the light-dependent reactions. And you end up with essentially a 3-carbon compound still, but it's different because each of these 3-carbon compounds now carries an extra hydrogen. So instead of phosphoglycerate, we end up with something called phosphoglyceraldehyde, or glyceraldehyde 3-phosphate. Some texts use this format. And there's still 12 of these puppies. They still have their attendant phosphorus. But like I said, they all have a hydrogen attached as well. All right, getting close to the end of the cycle here. And we left off with these glyceraldehyde 3-phosphates, these PGALs. What's really cool about these is that each of these is essentially a half of a glucose molecule with a phosphate attached. All we have to do is strip off the phosphate, rearrange some bonds, join them together, and you got a glucose. So what we're going to do is take 2 of these G3Ps, and we're going to go ahead and convert that into glucose, plus a couple of phosphates. Awesome. Only thing is we're not quite at a cycle yet, because now we've got 10 G3Ps, 10 PGALs left over, and we need to get back to our original ribulose biphosphate here. So we need another step. So here, final step here, to get back and complete this cycle. Remember, we left you off right about here, and after we remove our 2 PGALs, our 2 G3Ps, we ended up with, follow along, 10 of these G3Ps, right? Which will give us 30 carbons, which is what we need for our 6 ribulose bisphosphates, but only 10 phosphates, and we need 12. So we're going to have to get those phosphates from somewhere. Well, here's where they come in. We're going to take these smaller molecules and make a larger molecule. Well, anytime you go from a smaller molecule to a larger molecule, you're probably going to need some energy. Guess where you get that energy from? Yep, ATP. We still had 6 ATPs left over. So we're going to pump in 6 ATPs here. This really gets messy here. And when we do, we're going to use the energy in the ATP to build back our ribulose bisphosphate. What you end up with is 6 ADPs. But guess what? 2 of the phosphates get left behind, and you end up with 4 free phosphates. Why are the 2 phosphates left behind? Well, because over here, remember you had 30 carbons. That's a C. And only 10 phosphates. But we needed 12 phosphates for our ribulose bisphosphate, so hence 2 phosphates left behind. Now, if you're really wondering, well, gosh, aren't you losing phosphates every time you go through the cycle? Not really, because remember this puppy over here? We don't need these phosphates to make a glucose. And there's our 2 missing phosphates. We can pump that back into the system, and all is well in Calvin cycle. So Calvin cycles may be a little bit more complex than the light-dependent reactions and might take a little bit more practice. So kind of go back and dissect this step by step. Remember, it's really difficult to eat an elephant if you swallow the whole thing at once. But if you take it one bite at a time, it's a little bit easier. Do the same thing with those light-independent reactions, the Calvin cycle reactions. Take it one step at a time and see if you're able to go ahead and explain it piece by piece. If all else fails, get a hold of one of your bio teachers, make them sit down, and see if you can go through it, explain it with him or her. Anyway, best of luck. See you in cellular respiration.